Hi everyone, in this video we present J-EDA, the practical workbench for the analysis of diversified similarity queries about content-based image retrieval tasks. If you wish to use J-EDA, we have a demo version available on the link that appears on your screen. Before we demonstrate J-EDA, it is essential to note that J-EDA is not commercial software. It was designed for academic and demonstration purposes. When running J-EDA for the first time, it is necessary to configure the datasets that will be used by the system. This can be done in two ways. At first, the user must provide a name for the dataset and inform the directory where the images are. From there, J-EDA can extract the features of the dataset. In the second form, the user can provide to the system vectors of features generated by other tools. For this it is necessary to inform the name of the dataset, the type of the features, the directory of the images and the location of the file with the features. For each dataset, several feature vectors can be provided and, when adding all, the modifications must be saved. After you configure the datasets, the user can go to the main interface. This interface is divided into 5 parts: the dataset settings, the Explorer module, the experiment modules, and the analysis of results. In the Explorer module, the user can perform queries and test some J-EDA functionalities. Initially, one should select the dataset and provide a reference image. Then, select the query criteria and its parameter, the distance function, the feature extractor and execute the query. At this point, the result set will be displayed. The user can visualize the images on a real scale by clicking on the returned images while pressing the Control key and then clicking to see the selected images. Another interface will be displayed, and there will be a list of the selected images. Clicking on one of the images and this list will display it in the center of the interface. To use relevancy feedback techniques, the user must initially label the images. To label as relevant, to double-click left on the image, and to label as irrelevant, to double right-click. After labeling the recovered images, select the relevance feedback technique, and feedback the system. This will display a new result. The experiment modules automate the analysis of several query parameters, allowing several queries with the same parameters to be performed. In supervised feedback mode, the user has to label the recovered images and start the relevance feedback cycle. Initially, you need to select the query images that will be used during the experiment, select the query parameters, and start the evaluation process. When you start, the result of the first query will be displayed. At this point, the user can analyze the result set, label the images and feedback the system. As well, proceed to the next query image and repeat the evaluation processor, finalize the evaluation. When finished, the results obtained during the evaluation process will be saved in an external file, which can be used for later analysis. In the unsupervised feedback mode, the relevance of the images and inferred on the class of the query images in the result, that is, equal labels imply on relevant images. As in the previous modules, it is necessary to select the search parameters. Because of the unsupervised evaluation, it is necessary to provide the maximum number of relevance feedback cycles and to provide a file containing the classes of the images. Also, the user can select the query images in 3 ways. In random sampling, the user has to inform a location where the query images are and the number of images that must be selected. From this, the J-EDA generates a random list of images without repetitions. Already in the script selection, the user needs to select a file that contains the selection of query parameters and a list of query images. In manual selection, the user must select each of the query images. Also, if the user wants to use externally generated features and query images that are not part of the configured dataset, manual selection allows a file containing the characteristics and path of the query images to be loaded. In both cases, when experimenting, the results will be saved to an external file. The results analysis module is an easy-to-use graphical interface that allows the user to analyze the results of the experiments performed using metrics such as map. This interface is divided into 2, one for the supervised experiments and another for the unsupervised ones. Initially, it is necessary to select which evaluation metrics will be used and which results and files should be analyzed. After selecting the files, J-EDA will plot graphs with the results of each of the selected metrics. If the user wants to export the generated graphs, click Save Analysis and inform the directory where they should be exported. Thanks for watching this video, and try J-EDA.